Okay, so I just wanted to pick up right where we left off, except we're going to do a quick review about how the beginning, the first part of the urea cycle, and the fact that you start off with either glutamine from the extrahepatic tissues or alanine from muscles, which alanine gets transformed to glutamate by aminotransferase, specifically the alanine aminotransferase. I should say it's not alanine to glutamate, it's alanine to pyruvate. It's just the nitrogen gets added to alpha-ketoglutarate to make glutamate. All right. And so the glutamate can go ahead and enter on into the holy of holies of the mitochondria, the mitochondrial matrix. Glutamine itself can just go directly on in. Okay? So that's why it doesn't have to be modified in any way. The enzyme that removes that terminal nitrogen group from glutamine, once it's inside the mitochondrial matrix of the liver cells, called glutaminase. So now we have both species being a glutamate. And we talked about that there are two possibilities here for the glutamate. One is if we no longer need to make other amino acids or anything like that, then you can just go ahead and use glutamine dehydrogenase, which requires either NAD or NADP. And remember, it was special because it's the only known enzyme that it works with either one. It doesn't matter which of the two. And that would get rid of the N nitrogen, and then the carbons themselves are then alpha-ketoglutarate, which can go on either to the citric acid cycle or for the other, or to be used with other aminotransferases. Or the other possibility is what we've just been seeing, and that's aspartate aminotransferase, to where the oxaloacetate from the citric acid cycle is utilized to make aspartate. Okay, and aspartate actually is important for later on. We don't want to forget that. And that's why you can't really tell it because I've covered it up. But aspartate, remember, it can go back and forth between the holy of holies of the mitochondrial matrix and the liver's cytosol. Okay, but the nitrogen itself, this is the first official, I guess, step 0, if you want to think of it that way, of the urea cycle. It takes place in the mitochondrial matrix, and that's carbamoyl phosphate synthetase 1. So it's making carbamoyl phosphate. Okay, and this is the carbamoyl phosphate, which is actually the first official step of the urea cycle. It occurs within the mitochondrial matrix, and then this product, citrulline, which is actually an amino acid, it's just not an amino acid that's utilized to make a protein. Okay, same thing with ornithine. So ornithine is also an alpha amino acid. It's named after, does anyone know what it's named after? What's the ornith part? A prefix for, or a word for? Birds. Yep, ornithology. So ornithine is another amino acid. It's just not used to make our proteins. And so the ornithine gets modified to become citrulline right here. Now this enzyme has a very difficult name for me to pronounce. But what class or classes of enzymes could this be? Since essentially what we're doing is, if you look, it's taking ornithine, and the only difference between it is that it's adding this carbamoyl group, which is carbamoyl, or an amide group, to the very end of the ornithine to make it to citrulline. So what class or classes of enzymes could this possibly be? It's a transferase, okay? And so what is it transferring? Carbamoyl. And so the name for this one, I would have accepted a carbamoyl transferase, but they also call it transcarbamoylase, which makes me feel like I'm swallowing my tongue whenever I say it. Ornithine transcarbamoylase. But I would also accept ornithine carbamoyl transferase, because that is what it's doing. So like aminotransferases versus transaminase. Okay. So it tells you exactly what it's doing. It is transferring the carbamoyl group to ornithine, and the product is citrulline. And citrulline actually looks an awful lot like one of our other amino acids. What other amino acid does it look a lot like? It does look a lot like arginine, which is important in a moment. Oh, by the way, this is already available online. I did put the PowerPoint online. All right, so the next step is where citrulline then goes out to the hepatocytes cytosol. And it's the same enzyme that does two processes to where the intermediate does not leave, similar to the way that we had a conatase in the citric acid cycle. Okay, so we make this, it's another one that's hard for me to say, citrulyl AMP intermediate, which this is one time when we're activating it at the expense of an ATP. But instead of adding a phosphate, we're adding the AMP part. Okay, but it's still a great leaving group. And the aspartate or anaspartate, remember we made an aspartate up above. Now it may not be that exact aspartate, but it can go across the two membranes of the mitochondria. And now the aspartate actually comes in, acts as a nucleophile to kick off the AMP. So we make this really large molecule, relatively large molecule, compared to alanine and pyruvate and things like that, called argininosuccinate. Okay, so what class or classes of enzymes could this possibly be? Because we do utilize an ATP. So what are the possibilities here? A kinase, which what do kinases do? They add phosphates, or the synthetase. So which one would make more sense to name it? This is a synthetase. And so specifically, what would you call it? That is correct. This is argininosuccinate synthetase.
So that brackets is just the
Intermediate.
okay.
Mm-hmm. It does not, it's not leaving and you don't find it loose in the cell. To show you that it's not phosphorylating it, it's actually AMPylating it. No, adenosine monophosphorylating it. That just doesn't sound as good. But AMP is a great leaving group. Okay. Now, I really like the way that this book color codes everything because it makes it easier to see what's going on. In this color is the nitrogen that, or one of the nitrogens that we're trying to get rid of. Okay. And in the other color is the other nitrogen that's coming from above. So it originated from the aspartate. And then we have, in the light colored shading, something that's very, very important. It had been like the body, the carbon backbone of the aspartate. And we have in this third enzyme has a special name, but what it does is it essentially cleaves argininosuccinate into two molecules. One of them is arginine. So arginine is not only important for making proteins, but it's also important for the urea cycle, for nitrogen elimination. And it kicks off our good friend fumarate. Where can fumarate go? Where have we seen it? Where does it go? Citric acid cycle. So this is actually a way that it can be used to make more energy. It can go on in and join the citric acid cycle. Because remember, our whole goal here is we want to get all the possible energy out of our molecules as possible, in order to be efficient. So we fully oxidize the carbon backbones. It is our divine design. That's true. Faith integration. That should be like more double faith integration. Like brownie points, metaphorical cooking before it goes to Jackie. It's our divine design.
We catch those in one day.
Okay. Wow. So I will tell you that the third enzyme's got a special name. So like we have fumarase and aconitase and those. And so what do you suppose that this one special name is going to be? It is not arginase, but you're close. No, we already have fumarase. It is argininosuccinase. Exactly. Yeah. But you know what? If this was matching, I mean, it would be really easy because there's only knowing what you know about these that are specially named. They're almost always due to their cleavage of the products. But I have done matching in the past before, but not. Well, we did matching on the citric acid cycle.
Wow, that was the citric acid. Nobody knew where it started.
So, all right. And now the last step of the urea cycle is where water comes in and it kicks off urea to make the ornithine. Of course, ornithine then can travel back into the mitochondrial matrix. And so we get the fully oxidized urea, which, where does urea go? Yes, it is eliminated. It's the technical term for it, but yes, it does go out into your urine. Okay, and so that way we can get rid of those pesky nitrogens, get all of our energy. And this enzyme also has a special name. And so, well, so I could see ornithase, but that's not it. Oh, I think it's probably got a cool name too, since you've already guessed it once. This is arginase. So this is arginase. Because it's breaking apart arginine.
So that's just gonna keep going.
What?
It's just gonna keep going.
Meaning that, yeah, the ornithine then goes on to go back to become citrulline when it gets its carbomoyl group. Yep. And the cycle goes on. Hakuna Matata. Disney integration. Yes, Michael?
Why does it have to go back into the matrix?
Because the matrix is where it's going to be transcarbomoylated.
Oh, just for that first step.
Yes. Which I actually said that without stumbling the time. Okay, and in case you couldn't tell, the urea cycle and citric acid cycle are linked. I mean, it's pretty obvious with the fumarate, but fumarate can also be transformed into malate using fumarase. And malate, as we already know, and fumarate for that matter, can go back and forth between the mitochondrial matrix and the cytosol. Most of the urea cycle takes place in the cytosol, except for that first official step and the step 0. And so they call that the aspartate argininosuccinate shunt, because we can get aspartate from the citric acid cycle via what intermediate of the citric acid cycle? What's the alpha keto acid that can be used to make aspartate? OA, oxaloacetate. Oxaloacetate, which it's frozen up now. There we go. Oxaloacetate, using the aspartate aminotransferase, can make aspartate. Aspartate can go on. It gets, used to make argininosuccinate, kicks off the fumarate, which can go on back into the citric acid cycle. So that's why there are some books that have even referred to the Krebs bicycle, where they link the two, well, technically 3, since it's going to be 3 circles when you do the shunt together, but most books now just refer to the citric acid cycle and the urea cycle with the shunt in between. Okay. Are there any questions over this? This chapter actually wasn't really long.